You are now in the How to Use Genesys webinar, part one. Thank you so much for joining today. My name is Christelle Rabil, and I am the Genesys assistant at the Crop Trust. Um, Today's session is planned for a bit more than 2 hours. And what we are going to do today is we have a very ambitious agenda. And we are going to start with a small introduction to Genesys for those of you who are completely new to it. So a bit of background and history on what it is, when did it start. Then we're going to go over the homepage of Genesys. And I'm going to show you how to log in and create an account and why this is important to be browsing Genesys while you're logged in. Then we are going to have a look at translated Genesys, how to access these translations, how to send feedback about them. Then we are going to demonstrate and have a look at how to browse through passport data, how essentially to use passport data filters, whether they are manual or whether you are using AI for filtering in order to find the accessions that you are interested in, if you are a breeder or a researcher. Then we are going to have a look at how to request the material if you found some accessions that you want to order from the gene banks, how to do this directly in Genesys. And then we are going to have a look at some functionalities when it comes to passport data, for example, how to find similar accessions, how to see changes over time to the data. And then we're going to have a look at how to download passport data, the different types of downloads, and the data standard that we use. And this would be all about passport data, but the next demonstrations that will follow will be concerning trade data, subsets, the subsetting tool, and then we will have a question and answer session. So sit tight. And with that, I will directly jump into Genesys and introduce you a little bit on its background. I will swap my screen, just one moment. So Genesys is the world's largest portal to information about crop diversity conserved in gene banks. It is only concerned with material that is conserved in ex situ in gene banks around the world. And this site makes it easy for all gene banks with collections of plant genetic resources for food and agriculture to make their data publicly available. It's a centralized platform that aggregates this type of data from more than 500 different gene banks around the world, with over 4.4 million accession records at the moment in Genesys. A bit of timeline. So in 2001, the United Nations adopted the International Treaty on Plant Genetic Resources for Food and Agriculture, which acknowledged this need to have a centralized platform for sharing this type of data among researchers, breeders, farmers. And then in 2008, Biodiversity International, the Secretariat of the International Treaty, and the Crop Trust started developing Genesys. It wasn't called Genesys at first, but started developing this platform supported by a grant from the Bill and Melinda Gates Foundation. And then in 2011, the first Genesys portal launched, including information from 3 of the world's largest gene bank networks, which are ECPGR, the European network, CGIR gene banks, and USDA. A year later, in 2012, a task force, which later became the Genesys Advisory Committee, recommended that the Crop Trust becomes the legal entity responsible for the development of Genesys. And in 2015, a major new version of Genesys established a system of partner institutes to make their own continuous updates. And today, users and data providers continue to use this platform, again, bringing around 4.4 million accession information from more than 500 gene banks to people around the world. At the moment, Genesys is managed by the Crop Trust and funded. And this is only thanks to all the donors of the endowment fund of the Crop Trust, of the CGIR gene bank platform that ran from 2013 to 2021, and from different donors and the Crop Wild Relatives Project, and mainly the Federal Ministry for Food and Agriculture in Germany. So this is a bit of history. And now I am going to jump right to the homepage and guide you through the main structure of it, just so that you can get familiarized with it, and the different sections. How can you get oriented? How can you find things? So you will see when you are in Genesis, you first have this main menu that you will have access to from every page in Genesis. This will always return you to this homepage, this landing page, this logo button. And then you will find 4 different sections. The first one is everything related to data. So the first sub-menu item is passport data. Then you will see subsets and core collections. Then you will have a look at access to characterization and evaluation data sets. And at the end, you will have access to crop descriptor list. This is everything related to the raw data. Then in directory, you will see different pages that we created in order to aggregate those 4.4 million accession records into different, I would say, pages that are pre-filtered. The first being by crop, the other being by provenance of PGRFA from which countries, and the other is the data providers. So putting these 4.4 million accessions according to who provides the data for Genesis, or on a higher level of splitting this data up into according to the different networks that are sharing data in Genesis. So I'll have a quick look at how these pages look like. Highlighted crops. So these are the accessions in Genesis divided by crops, and you will have little introductions and overview summaries of the distribution of all the data on apple in Genesis and how many are tagged with the crop apple. From the provenance, you will see a list of countries and small note about provenance. So the provenance of material is documented at the level of individual accessions, and this means that where do the accessions come from, in the sense that where have they been collected from on the field, not which are the countries, so which gene banks that conserve these accessions, where are they located? So this is directly regarding the collected material on site, where has it been collected from? And I show you, for example, in the case of Azerbaijan, there are 23,000 accessions in Genesis that have been tagged as being collected from Azerbaijan, whether it's from um, any field or from an institute or from a market. There are different types of sources, but from as a country, um, this is what we mean by provenance. Then we've clustered the data as well as in the third submenu item according to the data providers. So this is about the gene banks. So this is about the accessions based on who shares their data and essentially who conserves them. This is including the CG gene banks, national partners, and regional gene banks. For example, Fiji. When you click on the data provider page, it will show you the specific views codes associated to this data provider, because one data provider can provide data on behalf of many different gene banks. In the case of CEPAC, that's only one. In the case of, for example, I will show you Embrapa. This would be many. So this data provider called Embrapa is sharing data on behalf of many different gene banks, and you can differentiate them by the name over here, but more importantly, by the views code, which is the code associated, a unique code associated to every gene bank around the world. Here you see a small overview of how many accessions belong to this gene bank within the data provider Embrapa. And if you click on the link, that would redirect you to the specific gene bank page, where you have so many buttons here to click. The most important one being, if you want to directly view the accessions, is this one. This will redirect you to the accession table, where there's much more information about the passport data pertaining to the specific gene bank within the MTRAPA data provider. Moving on from data provider, we also have aggregated data based on the networks. So many different gene banks can form together a network. And this can be an official network with a specific structure and management. For example, the CGIR gene banks or the European network or USDA. But they also can be networks pertaining to certain projects. For example, our very own project at the Crop Trust, the BOLD project. In Genesys, we decided to create for the different work packages of this project, different networks of gene banks, also for the Seeds for Resilience project. So this is a way to immediately access those, for example, project related data all at once. So we see here for the BOLD project and its first work package, we have all of these gene banks belonging to this project. And in one button, you can browse the accessions of all of these gene banks. And there are more than 50,000 accessions belonging to over 10 gene banks. So in the directory also, we have bibliography, but we will skip. The third menu item is everything related to reading material. I showed you already in the beginning of this webinar, the very first reading material, which is about Genesys, some timeline and acknowledgements. But there are also different sub-menu items that are useful for you. The main one being the recent activity, because this shows you what is the latest in Genesys, latest updates, activities, tools being released. Sometimes we do stories with our partner gene banks. And this is also something, the section, you have an overview of the section in the main landing page in Genesys, which is right here at the very top, recent activity. But you can also access, so this only shows you the first 6, the most recent 6 articles we have, whether you click on browse all posts or whether you click on resources, recent activity, you will have access to over 150 different posts. So that's the full article page. We get a lot of questions on how you can join Genesys, whether as a data provider or as a user who wants to log in. And we have for that a page on either becoming a registered user or becoming a data provider. And we also get a lot of questions from researchers on how to attribute or cite data from Genesys in your publications. And for that, in the resources section, we have a citation and attribution article that details the how to cite Genesys and also links you to the terms and conditions of use for information. There's a contact us menu, and this is where you get access to our help desk email. And the documentation, a very detailed article on how the accession data in Genesys is documented on every single descriptor, what it means, and what values you might be able to find in a lot of detail. So if you're downloading Excel files from Genesys and trying to understand the different descriptors, this would be your best resource, along with, of course, the official standard that I will be presenting to you later. The document that is this table is this article is based on already. And there are other items that you can have a look at. And then here, the fourth menu item is My List. And this is where you request materials from gene banks through Genesys, given that you already have some accessions in your cart. And we are going to have a look on how to do this, how to add accessions to your list, and how to request the material. This button right here is originally the login button. So let me log out and show you how it's supposed to be. So that's the login button right here. And I'm going to also show you, speaking of logging in right now directly, how you can register in Genesys. So how do you create an account, log in, and why it's important. So um, why is it important to have an account and log in? I mean, you can definitely browse Genesys without an account. I think most of our users already do that. And the reason why it's important, I want to highlight to you today why it would be helpful for you to uh, browse Genesys while having an account, is first of all, there are a lot of functionalities that are restricted to logged in users. There are a lot of reasons behind this, but you can still use Genesys basic functionalities like filtering passport data, having look at different visualizations, at the map, um, at images, um, all without being logged in. But there are some advanced tools like the Genesys AI that you see here, like the subsetting tool, like other tools, traits that would be preferable, that you'd only actually be able to use if you are logged in, and the same for actually downloading passport data. So I'm going to do a very quick filter for chickpea, for example, and show you if you want to download um, the chickpea data from Genesys. And if you click on this download button right here, both types of downloads are grayed out because you would need to be logged in to do that. So how do you create an account? By simply clicking on the login button. And you will see that there are two sections for logging in. So those are showing two types of login. You can either log in through your Google account without having to create a username and password in Genesys, just by authenticating via your Google, and this can also be other domains. For example, my personal email is at croptrust.org, and I'm able to use this authentication form without having a username and password. Um, Alternatively, you can create your own local account with username and password, the username being your email. And if you don't already have an account, there's the button right here on the login page, and you would need to provide an email. So I created just a test email for that purpose for today, and the password, and you have to repeat the password, and you have to write your full name and authenticate via the CAPTCHA that you're not a robot. And now we do the test. I should select all animals that have wings and create an account. Now your user account is created. And now, if you go back to Genesys and log in using the username and password that you just created, you would be able to see here a menu that has many different items and a button right there. We're going to go over them. So the first menu item is your dashboard. And from here, you will be able to manage your profile or change your password. Managing profile is basically deleting your account, having a look at when your account expires, what's the email, what's the name, or generating an API token if you're connecting to Genesys API. But also on the dashboard, you will have access to this requests menu, and you will be able to see the requests for material listed. Right now, there's none, but I will demonstrate to you when we get to that part the different options that are available on this request page. The other menu item is logging out, or here from here, you will access the translation. So you also need to be logged in to Genesys to access Genesys in either Spanish or French or Brazilian Portuguese. And you will see here this button enabling machine translation. This comes by default because the translations that we added to Genesys are mainly machine translated, Spanish, French, and Brazilian Portuguese with specific pages or paragraphs or terminologies that are curated by actual people who are our partners. And shout out to everyone who helped us with curating those machine translations over the past year. You can choose to disable everything that is translated, machine translated, and only display the curated translations, so the ones that have been reviewed by an actual human. This would, of course, limit your Spanish, French, or Brazilian Portuguese translations in Genesys. If you decide to keep this on, then you would get the full website and with all of its data and functionalities translated. But we will see later on how you can verify which is, reviewed by a person and which isn't. So we demonstrated how to create an account, how to log in, and why it's important. And now we're going to go directly, for example, to Spanish Genesys, since we're logged in already. You will see that your browser sometimes enables automatic translations. This is separate from the Genesys translations, obviously. We got that question before. I'm making sure right now that this is disabled. Otherwise, it would be in blue. That's how it works in Firefox. In Google Chrome, I think it works similarly. But right now, what I'm showing you, just so you know, is the Genesys translations, not the browser-generated translations. And now we're having a look at it in Spanish. And let's say I want to access the subsets in Spanish. There's a disclaimer right here in red that translates to English, saying that this information is automatically translated to English. You can click here to see the original one. And so I clicked here. This message has changed and saying now that this paragraph or this information right here is the original text that came into Genesys. And that you can click here again to toggle to the translation, but that's automatically machine generated. Why do we have this feature? It's because, as you know, sometimes machine translations can be really misleading or confusing or unclear. That's the downside of having machine translations. But the upside is we can have all of the website with all the data and automatically translated. So if you're a researcher and if you're browsing Genesys in Spanish and you're unclear about a certain expression, you want to see the original, you can just click this button that is available in a lot of different pages, not just the subsets in Genesys. And that's how you basically toggle between the different languages. I'm going to show you in French. And you can change here from the URL. If you notice, there's /fr. If you are in the same page and you want it in Spanish, then click ES. That's a little trick. And there's also the Brazilian Portuguese, PT-PR. And now reverting back to English, English is the normal one without any language letters in the URL, as you see. So of course, because the bulk of Genesis is machine translated, and because we know that a lot of you speak either French, Spanish, or Brazilian Portuguese, we've created a translation feedback form because we know that a lot of you might spot some strange translations or wrong translations, and you might want to give us this feedback for us to improve. This would be, we would really appreciate it because we are still working on improving those translations and adding more, hopefully, in the future. So we created a feedback mechanism for you that I will actually share in the chat, but also just plainly demonstrate right here while we're at it. You can share your email address at the beginning of this feedback form. That's completely optional. And we would only use it in order to get back to you with questions, if any. The next, section here is what's the original language. Usually it's English, but it could be French or Spanish or Brazilian Portuguese. And what did you translate to? So let's say I translated to Spanish. And what is the page URL where you spotted the error? In this case, let's say that I spotted an error here in the Spanish version of this subset that is machine translated. So I paste the URL here. So let's say that the original sentence is the last one. The germplasm of the subset is useful for research and breeding. Let's say this is where you notice there's an error in translation in the original text. You just paste it here in original sentence, and then you paste the machine translated sentence that contains an error. And here you can, it's optional, but you can give a rating on how bad is it really. It might be completely gibberish, and provide suggestions for the correct translation in your opinion and some notes, and then you click submit. Maybe it should be something else. And then you click submit. And this, we would receive this, and we are always monitoring this form, the responses to it, in order to improve translated Genesis. I see already a lot of questions in the chat from Afolabi. Can anyone create an account, or is authentication by the crop trust required? Otherwise, allowing unrestricted access would lead to data manipulation and Genesis. How do we prevent such risks? Yes, thank you, Afolabi. It's a very important question. So as a user of Genesis, not as a gene bank who provides data, anyone can create an account and then have access to the platform only to use certain tools like translations and searching through AI and downloading data. For that, we don't authenticate assign certain permissions. The users can just, like I demonstrated, just go ahead, create an account, and directly log in and start browsing the platform while being logged in. But you should note that the permissions for this type of account is very restricted, only to viewing Genesis, not uploading, not deleting, not updating data. I think what you are referring to is when the account is then granted further permission, such as updating on behalf of a gene bank or becoming a data provider and relating this account to a data provider or manipulating data. For that, this type of account or these types of permission need to be authenticated by the crop trust, because of course, otherwise there would be a lot of data manipulation. So for more permissions than what I just showed you, which is just browsing, yes, the crop trust does need to authenticate and assign. Our team assigns those permissions ourselves. But in terms of who can create an account simply and start browsing, anyone can do this, of course, and we do encourage it. We have another question. Can a private researcher or breeder get a germplasm through Genesis, or is it limited to public employees only? Yes, as a private researcher or breeder or student or literally anyone, you are able to request the germplasm through Genesis to different gene banks around the world. The ones that share data in Genesis for starters, and the gene banks that allow material requests to be relayed to them through Genesis. Now, whether or not those gene banks accept the material request or reject it, this is based on their own policies. They could be national laws regulating this. If they are international gene banks, there could be availability of material that regulates this. Um, so this would really depend. The role of Genesis and the material request is just to connect you as a breeder or researcher, private or not, with the gene banks. We are just a connector, a centralized place where you can request material from many different gene banks at the same time without having to go through 500 different websites. But that's where our role ends. And we're gonna go in detail later in this webinar. Um, Another question is, how does Genesis help in regulatory steps like declarations, PC, etc.? We're going to also go through this when we get through the material request part. And a last question I'm going to take now before moving on. Eurisco is a data provider for Armenia. Does it mean that there is no need to create an account for a country's gene bank? Yes, Alvina, I'm going to actually show you the Eurisco network, so which we access here from the directory and PGRFA networks. And you see here, Eurisco. And already you will see data from Armenia in this page. You already have data from Armenia in Genesis, it's true, on behalf, having been published on behalf of Armenia from ECPGR. So you don't need to create any separate agreement with the Genesis team. This has already been done for you on behalf of Armenia, I mean, through Eurisco. But you can still create an account if you want to browse through and request materials from other gene banks to to have them in your own collection, for example. Speaking of why we're here at this page, and because the next item on our agenda is browsing through passport data, I'm going to go ahead from this Eurisco page and click on Browse Accessions, and this will redirect us directly to the passport data browser, where we are going to have a look at filtering the passport data. So you see here, we are now only in the accessions that belong to the ECPGR network, and they are over 2 million. So this is almost half of the accessions in Genesis. I will demonstrate to you the filters by type. I will not go through every single one of them because this would take more than 2 hours, because there are more than 19 different categories of filters, as you see here. With every category having sub-filters right here. So I will just demonstrate to you the different types. The first one being multiple choice. So the very first filter you will see here, and by default, it is filtered as only no, as the historical accessions. So what you see also here as different filters, that what you see, the data is excluding historical, because only no by default is ticked under historical accessions. This means that you are only seeing active accessions in the Eurisco network as flagged in Genesis, not the accessions that have been discarded from the gene bank or archived or no longer existing. You're only seeing current ones. And this is why by default, you will see that historical accessions is filtered only for no. So not historical, current. You can always click here, toggle between or click both. And you will always see in these types of filters a small overview of the numbers. So you see here that over 2 million are marked as current. You see the same number here, but you also see here that around 20,000 are historical as part of the Eurisco network, which you will see if I flip the filters and click here, apply. This is crucial to click. After you apply filters, they don't automatically, after you click buttons, they don't automatically change the filters. You have to click this button at the end. And you see here now we are filtering only for historical accessions as part of Eurisco, and you see them all in red. The red color is only used as a hint that this accession is discarded. It's historical, it's archived. And we encourage you, of course, not to add such accessions to your list, and because you will not be able to request them from the gene bank. Please note that some accessions in the gene banks might be historical effectively, but in Genesis they do not seem they are not flagged as historical, and this is because of course there's a discrepancy in time between when the data changes in the gene bank and then until it reaches Genesis. So this is only a small disclaimer. So the next type of filter is text. It's a full text query. And I'm going to zoom in because there's a very important note about this full text search. So a full text search functions as it would in Google. It scans all of the data in Genesis, all of the accession records for a specific phrase that you are looking for. For example, let's say millet. So this is all of the accessions. This is prompting Genesis to look in all of the data, accession data in Genesis for the word millet. Now, this can be the crop name, but this can also be in remarks associated to an accession or in an accession name, or it might be someone's for example, if the collector's names are included, someone might have the family name Millet. So this is really very, very general. And this is why when you use this type of search, please keep in mind that you are not searching for a species or a crop only. You are scanning all the accession data for this phrase. Now, there are certain manipulations you can do to this full text search in order to make it even more broad. For example, if you want different spellings of millet, because sometimes it's millet or it's millets, and you want to make it more flexible, not to search for the exact phrase. So in this case, what you can do is using the symbol, and this would indicate to Genesis that you are searching for a prefix millet, but then any other word after or any other character after can follow. It's not specifically millet, just millet is the prefix of this phrase. And you can read this here in the notes. So I'm going to click enter. And you see here, Genesis found 1,000, more than 1,500, 400 accessions with this phrase millet as a prefix. And you see it's not necessarily the crop. I mean, it's different crops, Corso millet, common millet, um, Japanese millet, finger millet, but it's also scanning Genesis for this phrase in other fields other than the crop name. Another useful thing in this full text query is using double quotes. So if you want the exact phrase, let's say you want a very exact expression or word for example, a type of rice called ungu, then you put it in between brackets, indicating to Genesis you want this exact word, no more, no less. You can also use um, the vertical slash, this symbol right here, as an either or. So you either want millet or rice. So Genesis then would scan both, either or. And what's really cool that we don't have in the notes is if you want to search by an expression that sounds like something. So I want to search for anything that sounds like bungo. And then for this purpose, I write this expression and I put next to it this symbol. This means that I want Genesis to look for accessions that have anywhere in their data something that sounds like this. And you click enter. And this is a very, very broad type of search. I will remove the network ECPGR for that purpose. Or create, or for example, use dice. So I changed the full text query to search for something that sounds like dice, and there's different things, and there's dicentra, and different three different entries here, accessions that have in their data something that sounds like dice. I will remove this filter now. And then I will show you the next category of filters, which is filtering by holding institute. So I mentioned in the beginning that different gene banks are flagged. They have their own code that is assigned by the UN FAO, and this is called the views code. It is composed of 3 letters and 3 numbers, and this is unique to every institute. So you can search Genesis passport data according to institute codes, and this would mean that this is the institute code of the holding institute, so of the gene bank that conserves this material, not the gene bank that donated it or collected it from site. This is specific to the holding institute, which is the gene bank that conserves the material. And for that, I'm going to give the example of IITA that has the code NGA 039. Then a drop-down menu appears here. I click it. And once I click NGA 039 from the drop-down menu, you will see that there are 2 buttons next to it. It's now been entered in the filters pane with 2 buttons. The first one being plus and the first one being close. So this removes NGA 039 from this filters pane. And I want to add it again. And the plus button is an indicator that you're asking Genesis to look for accessions that are conserved in NGA 039 from all of those 4.4 million accessions. But you can also click on this plus button and it will transform into a minus. And this means that you are looking for all of the accessions in Genesis except for the ones conserved at NGA 039. And you can, of course, mix and match using these different exclusion or inclusion buttons. Let's say you want everything in Genesis that is all the accession records in Genesis except for NGA 039 and, for example, Icarda, LBM 002. Again, you have to turn it into a negative and then apply filters, of course. And you see the 4.4 has dropped to 4.2. Again, you want to remove those filters, so you can click the close button here and here. But we are keeping this one again because we only want to look for current accessions, not historical ones. So we are excluding the historical. You see under the field where you can type in your holding institute filter are some suggested ones with a breakdown of the accessions that they conserve. And this always goes in descending order from the highest to the lowest number. And then maybe you want to look for accessions that are conserved in gene banks in a certain country. So let's say that Nigeria has IITA as a CGIR center, but it also has the National Gene Bank of Nigeria. And you want to look for the accessions that are conserved in both. So instead of typing in here the code of Nigeria, NGA 039 and NGA 010, you can instead just click enter instead of clicking the drop-down menu. So instead of doing this and then applying filters, you can just type in the country code of Nigeria, which is NGA, and click enter and apply filters. And you see here, there are accessions from the institute code NGA 039. But if we go to the overview button, you would see that there are also accessions from many different gene banks in Nigeria that conserve accessions and share in Genesis that are NGA 010, the National Gene Bank, and other gene banks or universities. I go back to the manual filtering. And maybe I want the reverse if you want to look for all of the accessions in Genesis except for accessions that are conserved in Nigeria and maybe also in Germany. Then you make sure to include all these country codes and then turn the symbols into minus instead of plus and click on apply filters. And you see it's 4.1, almost 1 million accessions that are conserved in gene banks all over the world, except for gene banks in Nigeria and Germany. And from those accessions over here, maybe in a certain publication, you happened upon a specific accession number that you want to look for. We've had this question a lot from researchers that they read a publication or they are conducting research and they know very specific accessions that they are looking to request from gene banks and or having a look at their data. So this is where you can plug in the accession number. I will give you an example. One moment. For example, an accession from Erie, a rice accession from Erie that has the accession number IRGC 3209. And I have to click apply filters, and I have to click enter first, and then apply filters. And I will go to the accessions tab. And here I have filtered only for the specific accession number. Now, please note that this filter is sensitive to spaces. And so if you look for the accession IRGC without space 3209 Genesis will not result in any result. So, or alternatively, you can look for accessions, all of the accessions except this one by toggling through this number. If the accessions follow a certain sequence, consecutive sequence, you can say, for example, that you want the accession number that has the sequence number at least 200 and at most 1000 after a certain prefix, like IRGC is a prefix, and you want the sequential number to be a minimum of 200 and a maximum of 1000. And you see here, this has narrowed it down to 3.9 million accessions with prefixes minimum 200 and maximum 1000. And also this that takes into account the leading zeros at the beginning. So that's for the accession number filter that I can also remove from here. And then I will move on to the date search. So this is another type of search in Genesis that you will see on many different pages, not just Passport Data Browser, but this is basically everything that's on the left side is a minimum number, and everything on the right side is a maximum number. So this is how it works in Genesis, not just for dates, but also for, you see here, numbers and other values. So here on the on the left-hand side, there's a created on as early as, for example, the 1st of January 2024. And this created on filter means that this accession has been first published in Genesis on this date. Because there's a difference between created on and last modified. Last modified means the accession might have been there from before, but it was last updated. This data was last refreshed as early as the 1st of January 2024. So those are two separate meanings. And within the last modified, so when was the last update made, there's a specific filter we have for when was the taxonomy last modified. So maybe there has been a re-identification of certain accessions in a gene bank, and a rice accession turns out to be actually another species, and the taxonomy has been changed. So this filter will show you accessions with their last taxonomy modified um, at as early as the 1st of January 2024 um, as a minimum. So those are three separate ones. You can use them simultaneously or two at a time or one at a time, but just so that you know what they mean. And on the left-hand side, we are talking about um, the earliest date, and on the right-hand side, we are talking the latest date. So maybe I want to delete all of these here by pressing on the day and clicking delete on my keyboard. And maybe I want actually the filter for the accessions that have been created the latest at the beginning of this year, 2025. And I can leave everything else empty and just click on apply filters. And what Genesis in this case would filter out is all of the accessions that have been first published in Genesis from the beginning of the platform up until the beginning of this year. And this is where I uh, entered this state. And I click on apply filters. And again, I can remove filters from over here. The other category of filters that we already saw with historical is relating to the crop. So So this is a multiple choice filter. You can choose many at the same time. And also, if you double click on one of them, either one of them, the check button will turn into a negative, and this means you are excluding from your search certain crops. Maybe you just want all the others, excluding certain things, um, and then you can reset um, this filter by clicking one more time. You can untick by clicking one more time. And of course, never forget to apply filters. And of course, you see here a long list of crops. And you might wonder, for example, why aren't some of them included? This doesn't mean that Genesis doesn't have information about the crop that is not included in this list. It just means that um, you cannot search for this species using the crop filter. You will have to use a more advanced filter, which we always encourage users to choose instead of the crop filter here, which is the taxonomy filter. So this is the specific scientific name search that is definitely more accurate than the crop search in Genesis, because some gene banks might choose to upload fava beans accessions, but they don't tag them as the crop fava beans. So you might miss a lot of important material by using the crop, uh, but it won't be the case if you use the species, or at least that would reduce this possibility. So in the taxonomy search, you will see that the first button here highlights that by default, this search will, by a taxonomy, will include synonyms. And we use the GRIN taxonomy as the backbone of Genesis and the synonyms based on that as well. You can always disable it, but by default, it will be included. You can search by genus alone, for example, Triticum. Of course, you can either choose to include or exclude Triticum and remove the filter here of genus. You can choose also to search by species, so the full species name. For example, Hordeum vulgare. I'm clicking here from the suggested filters, but of course you can type in any species name that you want to look for here in the species. This is both genus and specific epithet. And there's just a category only for the specific epithet, which is just vulgare, or also excluding it. And you can also search by subtaxa and by GRIN taxon ID. Um, Another filter that you will see here is the origin of the material. And this is related to where the material has been collected from on the site. I will go quickly over it. Uh, you see here, there are also, in addition to this geographic provenance, there's also, I mean, in addition to this country-based search for the provenance, you can search for specific geographic coordinates, such as the latitude, longitude, and elevation. And as we've seen in the date filter, you will see a left side and a right side for every search filter category, the left side being a minimum value and the right side being a maximum value. For example, you might want to look for accessions that have been collected at a maximum of 500 meters above sea level. And please note that the here latitude, longitude are in degrees, decimal degrees, and the elevation is the unit is meters above sea level. We arrive here to another filter category, which is also a date, a specific date in this case, not a minimum and a maximum. And this is the collecting date. So it's the date from when the accession was first collected from the site, from the field during a collecting mission. And there are here some filter categories related to this collecting data, the collecting number of the assigned to the accession during this mission, the number of the collecting mission or its name, and the date. So you will see here that these two are text, and right here it's a number, but it's a different format. So it starts with the year, followed by the month, followed by the day, without any space. So, for example, if you want material that has been collected um, from the years 2000 and onward, then you would type in 2000 dash 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 if you don't know the month or the day. Or if you want a specific date, for example, the collecting date of 2019, May 1st, click enter. So that's a very specific day for the collecting. And you can also filter by location of collecting site. This is also text-based, and you can include specific area names or states or regions, even specific directions. The other way you can filter accessions in Genesis, which might be really interesting for you, is according to the biological status of the accession. This functions as a multiple choice also with with the same options about excluding and including, and also you can choose the many at the same time. So you will see that the different types are wild, and these are the subcategories of the wild material, then the weedy, then traditional cultivar or landrace, then breeding material with the different subcategories of the breeding lines um, or breeding type, and same for genetic stock with the subcategories, and also different biological statuses. Um, you might be interested in a specific wild, um, like semi-natural wild. You might only want this type, so you can just click on this or click on all of wild, which will select all of its subcategories as well, or exclude them as well. And as usual, you will see here a distribution of the accessions in Genesis that are already pre-filtered across the different categories of biological status as a quick overview. But we will see an advanced overview in a bit. Same type of filtering for the germplasm storage, whether it's in a seed collection, in the field, in vitro, cryopreserved, etc. And then you get to the status category of filters, which also functions the same way, but here you can get to choose to filter accessions that are only available for distribution. So you only click yes, and you see how many. You can get to choose accessions that are only georeferenced, so only accessions that include coordinate data, and accessions that are only included in the MLS, accessions that are only backed up in Svalbard, or the ones that only have images, yes or no. The curation type, if it's fully curated, partially curated, or archived as part of a CG gene bank, or if it belongs to the ages accessions. I will skip this for now, referenced accessions. It's a little more advanced, so I will get to it later on. What I also want you to have a look at and explore, which might be interesting to you, is that in addition to all of these filters that we've seen, which basically rely on the data as provided by the gene bank, we have an additional set of filters that we call climate at origin. And this is data that the gene banks do not provide. This is data that Genesis links to the accessions separately. If the accessions have geo-coordinates. So if the gene banks decide to share accession records with their coordinates of the collecting site, then Genesys gives you the option to filter out these accessions based on certain climatic functionalities associated to these geographic coordinates. For example, we have the annual mean temperature in degrees. We have the temperature seasonality, a maximum temperature of warmest month, etc. These are based on the world clim indicators, and they also function with a minimum and a maximum. For example, if you want to search for accessions that have been collected from an area that has an annual mean temperature at least a minimum of 15 degrees, you type in 15 here, or maybe you want a maximum of 15 degrees, then you type 15 here. And if you want a very specific number, which is 15, then you type it in both. This is when you want an exact uh, annual mean temperature, not a range or a minimum or a maximum. And we have 19 here different climatic indicators, I believe. Precipitation is included, annual precipitation and temperature that I invite you to discover. Before moving on, next I want to show you the different visualizations that we have. You've already seen the tabs here. I will have a look at the questions and answers. So someone is saying, if we used to have an account a long time ago and haven't used it, how do we recover it? Genesys removes accounts that haven't been used in a really long time. And in this case, you will need to recreate an account, especially if you haven't used it in more than 1 year, then you will need to recreate it again from scratch based on the steps that I demonstrated in the beginning. Someone is asking about historical accessions. Does it mean that the accessions do not exist anymore? And why do we not remove them from the database? Yes, this means that the accessions either have been lost from the gene bank, the material maybe died, or has been completely removed from the collection. The reason why we do not remove them entirely from the database is to keep track of what was there before. Also, in certain publications, for example, there might be links to these accession information. That's also interesting to keep. So we keep it just for recording purposes, for keeping this information for basically the purposes of keeping track of what was there before and keeping links to accession information that users might find interesting or useful. There are also researchers that do some analysis based on whether or not the accessions are historical. We keep that also for that purpose. Someone's asking if you cannot access Genesys unless you have an account. No, of course you can browse through Genesys without an account, but you will have a limited functionalities access. So you will not be able to download, for example, data if you don't have an account and use translations and other functionalities. So we find that it's a limited access. All right. I will continue then right now by showing you some overviews and some different visualizations of the data that we filtered. The first tab here being the overview tab. And you will see very colorful charts with many different ones. So the way this works is that this is following the order of the filters that you see here on the right, on the left side. the holding institute, the crop, the taxonomy, origin of the material. They're all the results are displayed here in these types of charts that you can copy and paste in Excel, for example. This would only copy actually the table in a tabular form. And it always ranks or lists the results uh, based on first the highest number of accessions in Genesis going down, whether it's the Holding Institute overview or the country of Holding Institute, the crop name. There's also genus name provided to Genesis, species name, um, matching GRIN taxonomy, biological status. So this is like a distribution of all of these 4 million accessions that we filtered for across the different categories, biological status or germplasm storage, etc. And using this button here, you can copy separately every chart, but in a tabular format, and paste it in Excel. I will show you how. So I copied and I'm pasting here. And there's the Genesis website, for example, for the genus. And all of the others. And you will see that there's many of them. There's also information including the provenance of the material, the site of safety duplication. A lot of it is in Svalbard. This is the use code of Svalbard, but also other sites of safety duplications, breeding institutes, status in the multilateral system, etc. Availability for distribution and curation type. So that's the tab that's concerning the overview. Then we have the tab that's called accessions. That is can either be a tabular view, like here, a table that includes only a limited amount of information. You get the full information by downloading, of course. This is only a few columns that show you the basic data. And you can download using this button here. But of course, we will go over the downloads later. And you can toggle here between the table view, which we are looking at right now, or list. We will go back to this table, but I want to also show you the next tab, which is the map view. This shows only the accessions that have geographic coordinates provided to Genesis. And it shows you how these accessions are distributed on the map based on their collection sites. And you will see here more buttons overlaid on the map that we will go through quickly. We have the zoom in, zoom out. We have the show climate button. So if you press on the show climate and press it on any point on Earth that is land, not on the sea, this will show you like an overview of the different climatic conditions, mainly temperature and precipitation, with different charts across the months of the year. And if you want to stop this functionality, you can just click cancel. And maybe you want to select a cluster of accessions that are only, let's say, in a specific area on the map. Then you can click this button here and you can draw a rectangle, for example, let's say here. And this would select only the accessions that are in the rectangle. And of course, you can reset or go back one step by closing the filters that have been created here by adding this rectangular filter. So in this case, if I want to erase this and go back to my previous map, I can just click on close for the latitude and the longitude filters, which is what I'm doing right now. The next button you see on the left-hand side is a print symbol, which means that you can download this view. You can even zoom in and download it. Let's say you want this view specifically. You have to click this button and then you select an image scale factor. And this the image scale factor, think of it as a resolution. One being the lower resolution and the two being higher, four being the highest. So you select the resolution that you would like and you click on download. And this would download this view in your files. Sorry. You can click here the close button and go to your downloads and have a look at what Genesys downloaded as an image. You'll notice when you zoom in and out, there's a scale here that changes. And the orientation of the map is always north, and you see here the north orientation, just for your information. On the right-hand side, there are two buttons. The first one is for the different layers that you can create, the different layers you can add to the map, and those are climatic layers. So you see there are different layers you can add concerning mean temperature or precipitation or a change or projected changes in temperatures and precipitations for the year 2050. Let's go for the simplest one, annual mean temperature. If you go to this category, you can see the legend of the map that will be added to your existing one here, and you can here toggle the opacity. So if you want it to be 100% visible or a bit more transparent, then you lower the opacity by clicking on the button and toggling left and right. And in order to enable it, you just click here on this button, and here you have a site of the origin of this layer. So I clicked on enable, and this added a layer of annual mean temperature with this legend here, and I can make it stronger or more transparent or lighter. And of course, then I can print this map with different resolutions, as I showed you. I can also save this map by clicking on the save button here, and by clicking on remember map, which I can name map 13 with mean temperature and save. And this would save those filtered accessions that are all the accessions in Genesys that have geographic coordinate, excluding the gene bank country, Nigeria, excluding historical in red. And I can access it here from the map list. And I can also change its opacity. Of course, course, alternatively, you can download the data from Genesys that includes these geographic coordinates, and you can open this file in a GIS software and do your own analysis. But these are just basic tools. If you don't have access to GIS software, that could really be helpful when you want to have an idea about these accessions relating to certain climatic conditions um, on the map. I will delete now this layer and disable the temperature, the annual mean temperature layer, and revert back to my original map. And I want to show you another thing you can do in this view is basically click on any one of those dots, which represent um, collection site. And you might see only one accession, but you might also see a list of accessions. For example, here, you see from the same collecting site, there's a list of accessions. Or maybe there's only one. Sorry, my cursor is moving the map view. And if you click on any one of them, this will redirect you to the accession detail of this accession specific one that we clicked on with the name, with the number IG 20036. And this will redirect you to the page of these accession details that really contain all of the passport data of this accession listed in this table with the taxonomy, session names, and any associated data sets, so characterization data sets that are in Genesys where this accession is present. If this accession also has trait data in Genesys, then they are listed here on its page. And you see trait data such as on color, on roughness, gloom color, etc. Some geographic information, some associated climatic information to this collecting site, and the passport data completeness index, which we will get to later in some metadata. I'll go back one step. Because the next tab I want to show you is the images. So in Genesys, gene banks can also provide images associated with certain accessions like the seed or images of the plant in the field. This will, of course, need some time to load because they are large files for the most part. More than 270,000 images of more than 200,000 accessions. So there might be more than one image associated to the same accession. And that's why you see here the different numbers. And those images, you can also see if you click on one accession number, you will see the associated images also as part of the page. I will show you the subsetting tool and traits later, but right now those are the different visualizations that you can have after using the filters in Genesys manually. If you have any questions so far, please type them in the Q&A section before we proceed. What we've seen so far is the manual passport data filtering. We will show you, I will show you quickly the Genesys AI, which is an alternative to manually filtering. And then this would wrap up our passport data filters. There's a question in the chat. Is this map in real time? Uh, no, the map is not of sessions is not in real time. It's always as of the date of last update of geographic data. So let's say a gene bank has last sent to Genesis geographic data in last year. So this is um, this would be the time. Uh, this would be the status in terms. So it's not in real time. Same for the temperature um, layers, temperature or precipitation. They're not in real time. So now I will move on to the Genesis AI, which, as a small reminder, if you're browsing Genesis right now, you would only be able to access it and prompt it if you're logged in. A small disclaimer, we cannot guarantee the total accuracy or completeness or timeliness of this information. We are using AI tools external to Genesis, and this is just a little reminder that you can always also switch to manual filtering at any point while using AI as well. And that we are developing the Genesis AI this year. We launched it last year and it's still under development. And we advise you to use complete sentences to say what you are looking for, as if you are talking to a person, not as if you are filtering data from here. So I can prompt Genesis AI, I am looking for rice accessions conserved in Erie that have been collected in 2000 in 2000. Now we'll go to the accessions tab. No, there are 0 accessions. So now I can just start a new search by clicking this button and say I am looking for accessions from Erie. And Genesis then AI says that the number of accessions are more than 130,000 accessions from the Holding Institute Erie. I can then further prompt it to say to narrow down accessions that have been collected from, let's say, only from the Philippines. And please note that I am adding to this prompt, to this existing prompt. So I am adding to accessions that have been collected from Erie and accessions that have been collected from the Philippines only. So this significantly narrows down to 4,500 accessions. And maybe I want to say, and Africa. So in this case, Genesis AI is replying that the request for search from accessions from both the Philippines and Africa is not clear. Please provide specific countries. So you see, it's a very dynamic prompting and the search results. You can always go back and forth and say, for example, I want accessions also from Nigeria. So you see here that Genesis AI has added to the original, to the first search Nigeria in addition to Philippines as provenance countries from the accessions that are conserved in Erie. So you can say, and I also want them to be available for distribution. And you see here, Genesys AI has added availability for distribution for yes. And these are, this is the results. And you can see those filters also here in the filters pane. This is all of them. And you can always switch to manual filtering and continue your search from there and toggle between back and forth between the AI and the manual filtering. Maybe you find it easier than to say, okay, I want also accessions that are georeferenced. You just do it from here and apply filters. And this further narrows it down, etc. So you can always reset your search by clicking Start a New Search if you want to start from scratch. And what I also want to highlight to you is because this is still under development and we really appreciate your feedback in order to keep improving Genesys AI. At the end of this Genesys AI panel, you will see a space here where you can give us your feedback. You can say this is not working well when I prompt, when I ask for accessions in Africa. Can you add, can you make AI do this? So, and then if you click on this light bulb button, this would send your feedback, and you see here a small message saying, thank you for your feedback. And we always monitor this form in order to improve Genesys AI. So I will choose right now a specific accession from here by clicking on the accession number has a link right here. So I'll click on it, and this would take me to the accession landing page. And if you're logged into Genesys, you will be able to see two buttons here, show changes and similar. Those are two functions in Genesys that we have for you. The first one is showing changes to the data, to all the data that you see here across time. We keep logs of those changes for up to 2 years. So if the accession acquisition date has changed or any of this information, the biological status, the taxonomy, you'll be able to see it on the, if you click the show changes button. And the other button is the similarity search that I will demonstrate actually on another accession. Also from Erie, also Rice. Sorry, from Simit. I will show you the changes from this accession because I know that it has recently been updated. And if you scroll, you will see that the type of germplasm storage didn't have any value before, and then it was added. And you can see also here the day when this change took effect. And you can also scroll to the status in the MLS. This wasn't part of this data before, and it was added also on the 20th of January. And then on the 7th of February, the accession URL was added. You might be interested in this information or not, but it's a tool that's made available to you. I will also demonstrate the other button that enables the similarity search, where Genesys can scan all 4.4 million accessions in the database and give you suggestions for accessions that might be the same, that are super similar and could be the same. So if you click this button while you're logged in, Genesys is saying it's looking for similar accessions right now. This will take a moment. And now the similarity search is enabled with a big disclaimer. It's only for demonstration purposes. We cannot guarantee that those two accessions are exactly the same in different gene banks. But we can only give you a close match or rate how likely is it. And you will see here in the match rating, 4/4 means there's a pretty high likelihood that those two accessions are the same, just conserved in two separate gene banks. The second row shows you that it is comparing one accession from Erie with this accession number in Erie to another accession in USDA with this accession number in USDA. And the next row tells you what are the matching data points between those two accessions. So we have the country of origin that you see here, that's Turkey. There's the species name you see here, Oryza sativa, and a bunch of other numbers associated that are found in the records of both accessions. The accession name, Skaliak in both cases. In the other identifiers, descriptor, they are also Kaliak, and the number is 57 on both sides. So but then again, the latitude and longitude are not exactly the same. So we suggest you use this with caution, but it's a pretty nice tool um, that can provide you with close matches to the accessions you are looking for. In case they are not available in one gene bank, maybe you can order them from the other gene bank. And then you can close this using this button, but you can also move on to the next closest match by clicking this forward button here. And you can also have an overview. So the next closest match is another accession from USDA. And the next closest match is only 3 stars out of 4, which is an accession from Brazil, from Embrapa. Your original accession, the one you are comparing, is always on the left-hand side. So you can close here. And we are back on the accessions detail page. So we are on this page right now. What you see here is a third button, which is add accession to my list. Now it's added. You see the number here is 13, 12, 13. And there are also many other ways to add accessions to your list in Genesys. I'll give you an example. I'm now filtering for Vigna, and I'm going to the accessions tab. And you see here, there's a My List column, and you can select manually different accessions, and you see here the number is being added. Once you are confident that this is the list of materials you want to request from the gene banks, you click on this My List tab over here, and you have an overview of all the accessions that you're about to request. You can also view them on the map if they have geographic coordinates. And you can click on this button, Request for Material. This will start a 3-step material request form. The first one being a review of the list again, but this time showing you which accessions are possible for you to order and which are not. The ones that are in green means that it's possible for you to order from the gene banks, and the ones in red means that you cannot for various reasons. Sometimes the gene banks don't have enough quantities. Sometimes they just simply are not including those accessions for requests, and other times the gene banks altogether do not enable material requests in Genesis. But the ones that are in green, you will be able to proceed with to the next step, where you enter your personal information, starting with your email address. And please keep in mind that all notifications related to this material request, whether it's from Genesis or from the gene banks, will be communicated via this email address that you provide. Um, so I'm going to paste here a test email address and a name and a surname. And here I should specify the type of request, if it's personal or if it's related to work. Let's say if it's related to work, then I have to provide the organization name, the organization's phone, address, and country. Let's say I am ordering on behalf of the crop trust. The phone is not required, but the address is. Let's say it's Germany, or actually Bonn, the city in the country of Germany. And another required field is the specifying the use of material. So whether it's for research or for other purposes. And it's required here to write at least a few sentences about why you are requesting this material, whether it's for research or for other purposes. So you can say here, we are conducting a study on rice varieties grown in the Philippines and in Nigeria, and blah, blah, blah. There are certain minimum numbers of characters. This is in order for the gene banks to really understand the purpose behind your request. And then you click on next step, which is an overview of this material request. And then you have to click continue, where Genesis will display a disclaimer saying that once you submit this request button, your request will be validated. So you need to check your email inbox and your spam folder for a validation message from Genesis. And that the email address provided will be checked against the treaty's EasySMTA database. So I'm clicking here, submit request, but I still need to validate it in order for this request to arrive to the gene bank. And in order to validate the request, I will have to go to my inbox, to the email that I provided, and I will receive an email from Genesis, which I will show you in a moment. And what I also did is going to my dashboard right here, to the requests menu, and I clicked on my requests for material. And this, because I did the material request while I was logged in to this account, I was able to see my material request here flagged as not validated yet, because I still need to receive an email from Genesis and click on this validation. This will take a moment. Okay, it's going to take a few more minutes. So in the meantime, I will demonstrate to you another important functionality of Genesis. which is the downloading of passport data. So we have users that are interested in the material itself who access Genesys in order to request material from gene banks, but we also have researchers who are only interested in the data and who want to analyze this data. So what I want to show you now is downloading different types of downloads, explore together quickly the Excel files, and go over the data standard that we use in Genesys for you to get familiarized with it. So after having filtered or narrowed down those 4.4 million accessions to, let's say, only the ones that are in the National Gene Bank of Ghana, which is GHA091, let's say I want to download all of this information. So what I can do is click on this accessions tab, and you see here the radio button that is in green gives you access to two different types of downloads. There's the quick one and the MCPD one. The download MCPD is all of the information we have about this accession, all the passport data information. And the quick download, you get to select specific columns if you don't want the whole bulk of the information. This is specifically useful if you're downloading more than 100,000 accessions or a large file. And if you only need, for example, the genus and the species name, and you don't need the rest. So you can click, if you're only interested in the taxonomy, on this quick download, which will initiate a download of a CSV file that you can open. And it would be in this format. You would need to then split up the data to different columns by using these steps in Excel. So this is the quick download. And now I will show you the full MCPD download by clicking this button. Genesys will then give you a small disclaimer on citations and terms and conditions, and then you can start the download. It will definitely take more time than the quick one because it's downloading now all the columns associated to the MCPD standard. And it's going to open when it's complete. You can also further narrow down, for example, to specific accession numbers. Let's say you only want accession numbers GH1, GH2, and GH3. What you can do here is type in GH1 in the accession number filter, GH2, and GH3. Apply filters. And here you can download the full MCPD, so the full passport data of these accessions, which is of course faster because there are fewer. And here we open the file, and we're going to explore it quickly together. It starts with the institute code of the Gene Bank, National Gene Bank of Ghana. Then it lists the DOI of these accession numbers, GH1, 2, and 3, whether they're historic or not. They're not historic. The genus and species names, everything related to the taxonomy, of course, that is available to us in Genesys. The associated GRIN taxonomy, acquisition date, acquisition name, country of origin, etc. You will see here that these labels here, the column labels, might be a bit unfamiliar to you, to those of you who are not familiar with the MCPD standard. So Genesys uses the multi-crop passport data descriptors in its latest version, version 2.1. And you can cross-check what those labels mean in the same file that you download from Genesys in the sheet titled descriptors. So this sheet, we encourage you to read it before you explore the MCPD file, so the full passport data file. We encourage you also to click on this link that is provided. This all gets downloaded automatically when you prompt an MCPD download from Genesys. So we encourage you to read through this to know what the columns mean, what the different values can be. And also there's a link to the documentation page that provides you with even more information and more detail directly in the Genesys website that I will show you here. This is the one we had a look at the beginning of this webinar. You can also have access to the standard not only on the Genesys portal, but also by having a quick look at the MCPD version 2.1. You will find it on the website of the FAO and on the CGIR website. And it's also available in different languages, English, French, and Spanish also. I'll take questions right now before proceeding. There's a question about how does Genesys help in regulatory steps like declarations and PC phytosanitary certificates. Genesys only connects researchers and breeders who are requesting the material with the gene banks that conserves this material. So we are only connecting the two sides, and then the gene banks take it from there. So then the gene banks get in touch with the people who made the material request and discuss the next steps about declarations, phytosanitary certificates, and all of that. This heavily depends on the country where the gene bank is located and where it is shipping to, where the request will be shipped to. So if it's within the same country, it's definitely easier, but Genesys is not involved in those. This is something that the gene bank later discusses with the requester. There's also another question. In accepted, do we must provide official document of receiving germplasm for the host institute? So again, if the material request is accepted, the gene bank then is the one who will get back to you, Kian, and they will ask for documents or signing certain agreements together or any other things they might ask. This depends on every gene bank, on the policy within the country and on the gene bank policy. So Genesys is not involved in this. We are only involved in relaying the request from you to the gene banks, and then the gene banks take it from there. What I want to show you, and maybe go a bit over time, is characterization data in Genesys. So if you go to this menu item, the third sub item is characterization and evaluation data sets. You will land on a list of morphological characterization or evaluation sets of data that are uploaded by different gene banks in Genesys, and those contain traits, relating to morphology or relating to resistance to certain disease. And those are by crop, that you would be able to also filter these lists of these trait data sets. You can also filter them just like you filter passport data using a smaller number of filters here on the left-hand side. So if you are looking for characterization data sets from, let's say, IITA or from the National Gene Bank of Ghana, then you have to here paste or type in the FAO code of the gene bank. I will share with you after this webinar where you can get these codes from. Alternatively, you can also click here on the data provider menu and choose, for example, let's say Africa rice. You might also want to filter by crop or by evaluation site, the latitude and longitude of the evaluation site. Um, for now, I will just filter through the rice from Africa rice. So Genesys is saying that there are 5 data sets of characterization and evaluation from Africa rice gene bank. If I click on any one of them, I will be able to access more detail, but more importantly, you will see here on this big green button um, that this data set is also available in a searchable form. So previously, if you've been familiar with Genesis a few years ago, these characterization data sets were available in Excel. And this is a preview of how they were accessible to you as a researcher before. It's taking a bit of time, but you could have also downloaded the Excel file from here. And what you would be doing basically is filtering through the data set either in a downloaded file in Excel, which is disconnected from the passport data in Genesys and from the descriptors. So you would have to then also download the metadata in order to understand these columns and separately download the passport data in order to understand the passport information of these characterized accessions. But as of 2023, we made this data available and searchable directly in Genesys by clicking this button here. You see, instead of downloading Excel, we are able to filter on the, again, left-hand side, the traits that are in this data set by specific data points. You see here, there's minimal passport data information that you can filter with, such as specific accession numbers, biological status, and whether the accession is historical or not. And then you will see every column, every characterization column here is included as a filter. And there's a link that gives you more detail on this descriptor, what it means, who created it in Genesys, which gene bank, and the different criteria. And you can also filter depending on the type of this observation. If it's a number, then you can filter by a minimum and maximum. These are mostly numbers. If it's a scale type of observation, then you can filter in a range depending on the scale. If it's a coded value, for example, a color or a shape distribution, then you can filter by clicking different categories. And of course, you have to apply filters at the end. There's panicle length, panicle attitude, and maybe I want only the panicle attitudes that are semi-erect to horizontal. So you see here the number is being further narrowed down of rice accessions that correspond to these observations. Maybe also I want to add a specific basal leaf CS color that I want it only green. And this narrows down to 14 different items that I can add to my list by clicking this button right here. Altogether, if I want to add them all at once, I click this button right here and I go to the radio button and click on add to my list. Alternatively, I can only add a few by clicking them individually, or I can choose to add all of them by clicking this button right here and going to the radio button and clicking on add to my list. And you see all 14 are now in my list. I can also do the same to remove them from my list at once. So I click on this button right here, go to the radio button, and click on remove from my list. And you see the number here is 0. If I choose to keep them in my list, I can then um, proceed to request the material from Africa Rice, as I showed you before. And you see here, all of them are flagged as available for distribution because they're all in the green color. So going back, um, I invite you to check out this characterization and evaluation page and filter through it and search in the different data sets for accessions you're interested in. This might be more interesting for you than only a passport data search because it has a wonderful amount of information about morphology or resistance. Um, if you have questions, please type them in the Q&A. I will wait a few moments for you to type in any questions you have. Otherwise, we will close this webinar for today and follow up with a part 2. Again, this was part 1. We had a very ambitious agenda. We were able to cover today a lot of it. We went over a bit of history concerning Genesis. When did it start? What it is? We went over the homepage. We saw how to create an account and how to log in. And we went over translated Genesis and how to send us feedback about translations. We went over the passport data filters in detail and whether it's manual filters or AI search. And we went over the different visualizations and how to reset or remove filters, how to exclude or include certain data points. And we went over how to request material from gene banks, what's the role of Genesis in it, and how to go about requesting the material and how to view the status in the dashboard. We went over the similarity search and how you can see changes that have been made to the accession data over time. And we went over downloading passport data, and we went quickly over searchable trade data. So I thank you all for joining. You will all receive, everybody who registered will receive a recording of this webinar in the next few days, and also an invitation for the webinar on how to use Genesis Part 2. Thank you so much, everyone. See you on the next one.